Parkinson's disease affects roughly 1 to 2% of the global population. It is the second most common neurodegenerative disease after Alzheimer's. Parkinson's is characterized by four main signs: rigidity, bradykinesia or slowness of movement, postural instability, and tremor. These effects stem from the progressive loss of dopaminergic neurons in the substantia nigra pars compacta. Motor symptoms tend to appear when roughly 50 to 60% of these dopaminergic neurons degenerate, which causes a 70 to 80% depletion of dopamine levels in the dorsal striatum. Unfortunately, there is no cure for Parkinson's, and the best treatment options typically only provide relief of some of the symptoms, with no reduction or reversal of disease progression. Oftentimes, the disease arises spontaneously with no inherited mutation. In some other cases, autosomal dominant and recessive familial forms have been identified. Researchers are currently studying the monogenic forms of Parkinson's disease to begin to understand how those mutations lead to cell dysfunction and neuron death, in order to identify the true causes of Parkinsonism and piece together the pathways involved. In the late 1990s, researchers noticed that many Parkinson's patients of various ethnicities harbored mutations in the Park2 gene, often with large deletions of entire exons, but also some point mutations. This Park2 gene contains 12 exons that encode a 465 amino acid protein called Parkin. Parkin is an E3 ubiquitin ligase with an amino-terminal ubiquitin-like domain and a carboxy-terminal ligase domain. The second gene that was identified in recessive Parkinson's disease cases was found initially by examining a large Italian pedigree, which revealed mutations on chromosome 1 at the Park6 locus. This gene encodes a 581 amino acid protein on 8 exons, coding for a phosphatase and tensin homolog P10 induced kinase 1, also called Pink1. This protein has a predicted C-terminal kinase domain and a mitochondrial targeting sequence at the N-terminus, suggesting that it is imported into the mitochondria. This supports other evidence that mitochondrial dysfunction is involved in the pathophysiology of Parkinson's disease. It was in the late 1970s that evidence linking mitochondrial dysfunction to Parkinsonism was observed. Accidental exposure to MPTP, a contaminant in the synthesis of MPPP, an illicit drug, was found to cause Parkinsonism and dopaminergic neurodegeneration. This was found to be because MPTP would oxidize to MPP+, a compound that is selectively taken up into dopaminergic neurons, causing inhibition of complex 1 in the electron transport chain. Complex 1 deficiency has also been observed in the brain, skeletal muscle, and platelets of sporadic Parkinson's disease patients. Mitochondrial DNA mutations are also associated with Parkinson's disease. In autopsies, neurons from the substantia nigra of Parkinson's disease patients harbor high levels of DNA mutations in the mitochondria, including large deletions that cause mitochondrial dysfunction. Further, people with mutations in DNA polymerase gamma, the one responsible for mitochondrial DNA replication, accumulate excessive mitochondrial DNA mutations and have an increased risk of developing Parkinson's disease. These links between mitochondrial dysfunction and the pathogenesis of Parkinson's disease sparked interest in the role that Pink1 plays in the mitochondria. While Parkin resides in the cytosol and Pink1 localizes to the mitochondria, studies in Drosophila revealed that both these proteins function in the same pathway to maintain mitochondrial fidelity. Studies revealed that when mitochondria are depolarized, Parkin is recruited from the cytosol to the mitochondria, where it mediates mitophagy, or the selective autophagic removal of the damaged mitochondria. When mitochondria are dysfunctional, Pink1 accumulates on the mitochondrial surface, and its kinase activity is needed to cause Parkin translocation to mitochondria and cause mitophagy. These findings have further been developed to show a more detailed mechanism. First, Pink1 detects mitochondrial dysfunction. It then signals Parkin to ubiquitinate specifically the damaged mitochondria. This instigates the removal by autophagy. Thus, Pink1 and Parkin function in a sort of mitochondrial quality control capacity. This is why their dysfunction contributes to Parkinsonism. Mutations often inhibit Pink1 recruitment of Parkin to mitochondria, thus leading to the lack of removal of dysfunctional mitochondria. Now let's take a little bit of a closer look into this mechanism. How exactly does Pink1 detect mitochondrial damage? In healthy mitochondria, there is a rapid and constitutive degradation of Pink1, acting as a sort of damage sensing mechanism, whereby when a mitochondrion becomes impaired, Pink1 is no longer degraded at the same rate, causing its accumulation on the outer membrane of the dysfunctional mitochondria. In normal steady-state conditions, Pink1 is imported through the Tom complex of the outer mitochondrial membrane and into the Tim complex of the inner mitochondrial membrane. There, it is cleaved initially by the mitochondrial processing peptidase, MPP, and further cleaved by PARL, which causes the release of a 52 kilodalton N-terminal deleted form of Parkin back into the cytosol, where it is degraded by the ubiquitin proteasome system. When mitochondrial import through the Tom complex is disrupted by mitochondrial depolarizing agents, electron transport chain inhibitors, genetic and environmental stressors, or unfolded proteins, Pink1 cannot be processed by PARL because import into the inner membrane, where PARL and MPP are located, is blocked. Instead, Pink1 accumulates uncleaved on the outer mitochondrial membrane, bound to the Tom complex in a 2:1 molecular ratio. Taken all together, this mechanism outlines how Pink1 senses mitochondrial damage by regulated proteolysis. This shows how a single damaged mitochondrium would be flagged by Pink1. As mentioned, Pink1 has a kinase domain. Specifically, Pink1 is a ubiquitin kinase. Pink1 directly phosphorylates Parkin at serine 65 within the ubiquitin-like domain. This phosphorylation stimulates Parkin's E3 ligase activity and recruitment to mitochondria. Activated Parkin then ubiquitinates substrates on the outer mitochondrial membrane. These newly ubiquitinated proteins now also serve as targets of Pink1, which also phosphorylates ubiquitin molecules. These phospho-ubiquitin proteins can also activate Parkin's E3 ubiquitin ligase activity, although the mechanism is not fully understood. And thus activating more Parkin, this leads to a cycle of strong positive feedback, assuring the clearance of the damaged mitochondria. To simplify, we see that the phosphorylation of ubiquitin at serine 65 activates the ubiquitin ligase activity of Parkin. This ubiquitin can be on Parkin itself in its ubiquitin-like domain or on other mitochondrial membrane proteins. Once activated, Parkin modifies many cytosolic and outer mitochondrial membrane proteins by adding K48, K63, K6, and K11 linked ubiquitin chains. K63 linked chains appear to be involved in the recruitment of P62 and autophagy receptor. K11 and K48 linked chains on mitochondria likely lead to an endoplasmic reticulum-associated degradation-like extraction of proteins from the outer mitochondrial membrane by P97, VCP, and subsequent degradation by the proteasome. This proteolysis and removal of outer mitochondrial membrane proteins seems to be necessary for mitophagy. The levels of those proteins also control mitochondrial motility and dynamics throughout the mitophagy process. Till now, 36 substrates of Parkin on the outer mitochondrial membrane have been confidently identified. This may suggest that there is not a specific target required for ubiquitin signaling of mitophagy, and that general chain linkage and density on the mitochondrial surface may be the trigger. So now that we know how Pink1 and Parkin interact with the mitochondria to trigger mitophagy, let's take a look at the actual mechanisms of mitophagy. As mentioned, it is presumed that the ubiquitin chains that Parkin attaches to mitochondrial proteins initiates mitophagy. This signals the recruitment of three components of the autophagosome targeting machinery: a serine threonine kinase, UNC51-like kinase 1, membrane-spanning ATG9 protein, and LC3 associated isolation membranes. The recruitment of these and other proteins begins to form an isolation membrane around the tagged mitochondria termed the autophagosome, which is a double membrane structure. The autophagosome then fuses with a lysosome. As the lysosome contains many lytic enzymes, the contents of the autophagosome, in this case the mitochondria, are now exposed and digested or degraded. Thus, the damaged mitochondria are removed from the cell. As we've seen, Parkin helps clear damaged mitochondria in the cell, but the removal of mitochondria leads to another problem. Mitochondria help fulfill the energy requirements of the cell, and thus removal of a mitochondrium has to be compensated. It was found in 2011 that Parkin also ubiquinates a transcriptional repressor named Paris, causing its degradation by the proteasome and thereby inducing mitochondrial biogenesis. Parkin has also been found to accumulate on intracellular mycobacterium, inducing their autophagy as a defense mechanism. This is consistent with the fact that Parkin mutations are linked with increased susceptibility to leprosy. But Parkin exists in an auto-inhibited state and requires phosphorylation to its ubiquitin-like domain to be activated. How this is achieved in these other roles of Parkin remains unknown. So what's the real link between mitochondria and Parkinson's? How could reduced mitochondrial function lead to the symptoms involved in Parkinson's? Firstly, it's important to note that the real cause is not well understood. Findings in Drosophila don't always agree with findings in mice, and there is a wide body of sometimes contradictory evidence. Correlations have been established, but a precise mechanism by which reduced mitophagy leads to Parkinsonism is not developed. However, here we'll see the overarching hypothesis. As we know, many neurons have very high energy demands. Action potentials are no joke. Specifically, in the substantia nigra par compacta, the dopaminergic neurons have very long, extensively branched axons with many neurotransmitter release sites. These neurons also have a natural pacemaking activity. In other words, they're always active at a basal level. This pacemaking is driven by rhythmic movement of calcium into and out of the cell. While the intrusion of calcium into the cell drives ATP production, the extrusion of calcium counteracts that, sapping energy supplies. As such, these dopaminergic neurons are said to operate at a delicate equilibrium where further stress or environmental demand may lead to their degeneration. This is why age is a risk factor for Parkinson's disease, and it can arise without any inherited mutations. Regularly, when mitochondria become dysfunctional in cells, they undergo mitophagy through the signaling of Pink1 and Parkin, and the cell also simultaneously signals for the biogenesis of new mitochondria. So, with mutations in Pink1 and Parkin, as mitochondria lose their function, they are not cleared, and the cell does not signal for new mitochondria to the extent that they should. Thus, since the mitochondria are the powerhouse of the cell, generating the ATP needed for these dopaminergic neurons to function, these neurons no longer have their energy needs fulfilled, leading to the death of these dopaminergic neurons. The substantia nigra is thought to be heavily involved in movement, and so the loss of neurons from this specific area leads to the motor defects observed in Parkinson's disease, including tremors and bradykinesia.